Uh, you know, drug discovery. I mean, Dr. Raj Manar and also Dr. Ramesh Panchangula gave a very good overview of what happens to a drug that actually goes into development. I mean, it's not an easy job for pharmaceutical scientists. And on the other hand, discovering a drug is a pain, a real pain. I mean, I've been in the industry for 30 years now. The so the drug discovery and development, people think it's a checkbook exercise. Yeah, okay, this one, this one, this one, this one. What you do is checking boxes, and that is what many people do the mistake. It's not a checkbox exercise. Every drug has already made a point by Dr. Rajmanar that is different. It requires a different skill set. And today, even FDA is saying that modernize your drug development. I mean, we've been doing drug discovery and development for 100 years now, more, and we just use animals. Animals, animals, animals. But the problem comes is that are these animals really telling you the story you want to when you want to make a drug into the market? So I'll take you to a different thought process here. So I don't want to go into this because history of drug discovery, I think most people know about it. But today, with recombinant technology, genetic manipulation, organism microfluidics, 3D bioprinting, all kinds of gene editing, all kinds of techniques today are available. And FDA is saying use those technologies to make drugs, not the animals' old-fashioned way. It's a long process. I'm not going to talk about everything here. It takes 10,000 to 20,000 drugs to make one drug, and it takes 10 to 14 years, and anywhere between $4 to $6 billion to make one drug into the market. But the real bottleneck is clinical trials. It's not discovering the drug in early discovery and doing the animal toxicology studies, but 95% of the drugs fail in clinical trials. When you take a molecule into the clinical trial, you'll be surprised. Oh, my drug is safe in animals. My drug worked very well in animals, but you see failures there. So this is the valley of death. And if you look at the historical perspective, in 1996, 53 drugs were approved by FDA. Look at 2023, 53 drugs were approved by FDA. There's no change. Our cost is going up from $800 million from 1993 to today to $4 billion. Well, where are we going? Because there's no new drugs coming out. We have changed the technologies. We went from small molecule to biologics to all kinds of orphan drugs and unmet needs technologies we've been using, but we still don't have a success. So did technologies really help us a lot? No. I'm running a little bit fast because he wants me to finish in 30 minutes. So, in a drug discovery and development, a drug must be safe and it has to be effective. So, any drug that works but is not safe, FDA will not approve it. So, FDA actually regulates safety. Efficacy, okay, moderately effective also, they can approve it. But the safety is the major point. They don't want people to be dying with taking this drug. When you're doing the standard process in safety assessment, that there are lots of things that you need to do. Carcinogenicity studies take 2 years. Of course, you don't do for every compound, but FDA requires all these things up to 12-month studies. I'll bring you one point here. A toxicity may occur after 12 months. I'm doing pharmacokinetics in a single dose. So, single dose pharmacokinetics, how can it predict 12-month toxicity? Same thing with efficacy, right? How can a single dose PK can predict pharmacodynamics after 12 months? This is what we're not doing because antibiotic, it can do it. The reason is because antibiotic kills the bacteria and you can get a real PKPD, but you cannot get a PKPD for a cancer drug in one day or 1 week. You need multiple dosing for months. So the question you have to ask is, why do drugs fail in the clinic? In 1960s to 1980s, it was the pharmacokinetics. 40% of drugs failed. And then we put a lot of models, mouse, rat, dog, and monkey, pharmacokinetics, doing the allometric scaling approaches. We tried to predict human. But even then, we had only 30 to 40% success in predicting human pharmacokinetics. then we at least managed to do it. But if you look at this, efficacy failures were about 30% in those days. And then we managed the PKs, right? When you increase the pharmacokinetics, automatically the toxicities were going because you have more bioavailable drug. But the efficacy didn't alter, right? So we have a lot of tox models. Even artificial intelligence is now trying to do in silico models and everything that are trying to predict the toxicity at the moment. All those models also reduce, but now what you look at is in phase 3 trials, efficacy failures are 66%, which means that a drug that worked in an animal models in early on, when you go all the way to phase 3 trials, is not working. So you wasted $2 to $3 billion developing the drug. So how do we manage this cost effectively? You know, this chart I put it up every time because humans are not animals. Our animals are not humans. Basically, you look at major enzyme cytochromes P450s that metabolize the drugs are quite different from animals to humans. I'm not going to go into details. Species differences between mouse, rat, dog, and monkey and human sheep mediate drug metabolism. There's a lot of literature there. Species difference, pharmacokinetics and pharmacodynamics. Uh, Mostly here, it's a physiological differences in, especially in humans and animals. Drug toxicities, it's well established that same drug can be differently toxic if you take Sprague Dawley rats versus the Wistar rats. Not just a rat versus dog, even different strains have different. So, from 1980s on, there are always mechanistic studies were required to understand why the drugs are toxic or working by a mechanism, pharmacodynamic effect. So in vitro alternatives are not new. They've been there for 19. So in the beginning 1980s, we used to use subcellular fraction. Take a liver, grind it up, take the microsomes or mitochondria or nuclear fractions, and look how much drug is transformed. Now the technologies are evolving. We went to 2D cells, take hepatocytes or something, and and also recombinant enzymes. In 2000s, we went another step ahead into 3D cells, regenerative cells like you use induced pluripotent stem cells or mesenchymal stem cells, etc. Coupled to these things, you also have now microfluidics and 3D bioprinting technologies. The advantage is that when you take a single cell, it's isolated, it's not communicating with anything else. Liver has 5 different cell types. Okay, even they communicate with each other. So I take only hepatocytes and do the toxicity study or DMPK studies. They're not relevant to the human situation. So I need to bring the physiological system in place. So now with the multiple organ interactions, pleiotropic effects, clinically relevant mechanisms can be used through the microfluidic mechanisms. Not in even 3D cells on tissues, if you have only one type of cells, you still cannot do it. So this is one of the papers we published. Metabolism. You know, metabolism is not simple. Uh, because of these metabolites are forming, the drug is actually eliminated very, very fast from the liver. And so we could not get a very good efficacy for these drugs. Same thing for tramadol. Uh, we published, there are lots of enzymes. You know, cytochrome P50 is not one enzyme. There are 5 or 6 enzymes working together. Some people say, hey, I'm doing CYP26, it's a polymorphic enzyme. I'll look at the polymorphic pharmacogenetics. I said no, because it's also metabolized by CYP3A4 into CYP2C9, CYP2E1. So it's not one drug, one enzyme it's metabolizing. It's 5 different enzymes are metabolizing. Then you, if it's only CYP26 is metabolizing, then you need to look into polymorphic metabolism. And we also, in early 2000s, we got this high resolution LCMS system, Orbitrap, and we could actually, without NMR, we could characterize these metabolites. Carvedilol, which only had 5 metabolites early on, we could fish out with the high resolution mass spec about 40 metabolites. So how many metabolites you're missing? That is the problem. That what we have is the technology was not sufficient enough to be able to predict what we want to predict in humans. So the thing is, today we have technologies that I can fish out 40 metabolites Some of the metabolites are different from animals, and those are the ones that cause toxins. And the in silico alternatives. First started Derek, one of the toxicology software in 1992. We used that in silico software. Derek came and we actually had lots of drugs that failed. We looked for mutagenicity assays in those models. And since then, there are so many softwares that came in for toxicology, efficacy, medicine chemistry, all kinds of things that today, this I published in 2012. Today is more, and I think Dr. Sridhar can tell you more about it because he's right now focusing on artificial intelligence to discover more new drugs. So the softwares are ever increasing. So my story took a different turn. From I filed probably close to 100 INDs in pharma companies for over 20 years. About 5 drugs are in the market, but still, we success rate is very poor. Something called Dawn of the Zombies. I'm going to give you a little bit. This was published by Robin Orwant for the work we did in J&J in collaboration with Hurel, where we actually created a human on a chip model. This was in 2001 to 2006 that was done. It's not to replace the animals, but to design clinical trials better and getting a much better success rate in the clinical trials. So basically what happens is that you have a microfluidic chip, you have different tissues or cells in different compartments, and it is circulating. And this is how you develop model. This was very prototype model we developed, 2001 to 2006. I have to tell a little bit of story about Dr. Les Bennett, worldwide renowned pharmacokinetics, actually came to me and said, Manny, we need to do this. So I funded that project with Hurel, and I actually got this done. We got very primitive data, but we got a very good data here. If you look at, take the hepatocyte, human hepatocytes, put in a collagen-based matrix, and look at the cytochrome P50 enzyme activities. Look at the light blue bars, very low activities. But we did it in a circulatory fashion with other cells and tissues because nutrients, metabolites are recirculating. You see the activities are much higher up. This is just to give you a better idea that you need a circulatory system with all the organs communicating with each other. And I left the J&J, but I still have that passion to build those models. So I collaborated with Dr. Uday Saxena, my partner, co-founder in Reagene Biosciences, that we started the company. And we developed the 3D bioprinting approaches. Microfluidics is a little bit difficult. You need macrofluidics. Macrofluidics, you only put 50 cells into it, and analytically challenging, very challenging. So I want to do macrofluidics, but we don't have the chips yet. So we have 3D bioprinting. These are inkjet printers, and you put the cells on biomaterials. I'm not going to go into the details, but you can look at morphology, drug-induced toxicities, and even efficacies that you can analyze. So this is the process that we have developed at the Reagene Biosciences. Anybody who have questions about these technologies, you can come to me later. Okay. Here, what we did was we put intestinal cells. When you give oral drug, right? First, it is exposed to intestinal cells. Then it goes through the portal circulation into the liver, right? These two are very important because if I can directly go into the cancer cells, I'm getting wrong data. So drug transport, drug metabolism are addressed here. And then we are putting target cells, in this case, lung cancer cells, right? So we are actually mimicking the human environment stepwise, how the drug flows in the body. And we have a long way to go because we also have to put kidneys and other organs as well. But this is a prototype model that we already built. You can measure the cell morphology, drug activity, drug metabolism by LCMS, cell viability. You can use up to 96 well formats. We have tested up to 24 well formats. And we created a lung vascular model. We use this for COVID. We repurposed drugs for COVID and protein kinase inhibitors and anti-diabetic SGLT2 inhibitors really did well. To our surprise, actually, by the time we finished this project, WHO approved ibrutinib, one of the protein kinase inhibitors, as an anti-COVID drug. So indirect validation for our model. So to date, we developed vascular lung, cancer in a dish, leaky gut, liver axis, diabetes in a dish, 3D human skin. So this is the vision that we all have, that you have a human on a chip. But that's going to take some time. It's not going to happen tomorrow. And the idea is that you can use all these physiological parameters: blood flow, protein binding, adipose absorption, muscle concentrations, drug metabolism, drug transport, histopathology, drug efficacy, and you can create a PBPK, PKPD model using this model. And the vision is that export all this data to the in silico and AI models. I want to point out here that today AI models are rich in animal data. They don't have much human data because most drugs go into the clinic and fail anyway. So you don't have human. So what we want to do is enrich with these models the human data. So that can be transferred back to the AI ML models, and we can enrich the human data so that you actually predict what's happening in humans, not the data taken from animals. You won't, again, you're back to square one. You won't be able to predict what happens in humans. So this is PPPK models are not new. In 1990s, we applied for benzene toxicity. So there are lots of models that are being validated for that. So this is the kind of vision that you can combine all these things integrated and build a PPPK model. And you can also use that for accurate efficacy evaluations. So what are the major gaps in India? Reagents to be imported from the West. Primary cryopreserved human cells need to be imported from West. Biobanks of human cells are not available in India. Sophisticated analytical tools like high resolution LCM SMS are too expensive for a startup like me to buy them. Scientific experts to carry out these models are limited and need to be trained before embarking on each project. Filing global IP is time consuming and expensive process. Venture capitals and angel investors need revenue flow. They say, oh, how much revenues are you getting? These are innovations. You can't just get revenues right away. So what we are suggesting is propose strategic partnerships between Indian pharma and innovative startups. I've been trying this. Raj Manar is here. I'll ask him to volunteer me for this. Take it to Sun Pharma. In the US, what happens if I have a startup company and develop these models? The first persons I go to is J&J, Pfizer, Merck, Eli Lilly, and all Novartis, etc. And they give me $1 million each, and they actually try to validate, cross-validate, not just validate for one lab, cross-validate in multiple labs, and that then they write a white paper. That white paper is published. FDA looks at it, forms a guidance, regulatory guidance. We don't have that kind of system here. So, Rajmanar, be my guest. Take it to Sun Pharma and see if we could collaborate with them and validate our models. Yeah, absolutely. Right? And we could also share the IP and resources between startups and pharma. Okay. I've got lots of people to thank here. The major person is Uday Saxena. Uday and I did PhD together in Canada. We were roommates fighting with each other all the time. But even today we fight with each other. But, he is critical in developing these models for us. And we have a lot of these interns and who work with. I am also indebted to Dr. Fred Tonelli. I started working with him in 1992 at American Cyanamide Company. He single-handedly, only me, he took me to Purdue Pharma as a group leader, then as a global regional head of the drug development at J&J. So he and I clicked very well together, and I think he's one of my godfathers. So you need godfathers to be able to be successful in life. And Jose and Heng Kiang Lim, they're also very good friends and helped me in a lot of these projects. Okay. I also have to thank Professor Les Bennett, who introduced me to this 3D world, and Robert Freeman from Hurel. And by the way, Hurel is sold for about 70 or 80 million dollars to Matek, another company. So when you develop these tools, you're aiming to sell your company for $100 million, not just make it 2 crore revenue or 1 crore revenue here, right? Think differently. You're not making revenues from 100 rupees to 200 rupees. You look at, as you already mentioned, US companies investment, GLP-1 is going to make $100 billion. One drug. Our how much revenues are we making in pharma today in India? $130 billion. One drug is making in the US that kind of revenues. Think different. We need to be innovative, not doing the CRO mindset or doing the generic mindset. So please take this home for the message. And anybody who's interested in collaborating, please let us know. Thank you.